Our final speaker for this morning's really interesting session, representing New South Wales DPI, is Dr. Barbara Brito. I've known Barbara for 5 years, and it's been fantastic to get to know her and also to get to know her research, which Ian has alluded to in the first session. So here's the topic: when bugs go bad, who is attacking your calves? Barbara, welcome.
So thank you very much for giving me the opportunity to present the results of this project that we've been working on for the past 4 years. And it's mostly about bugs, but first of all, let's talk a little bit about calf health. That's not only bugs. If we want to keep a calf healthy, there are several factors, key factors that impact that calf health that will maintain it stronger and that will prevent it having those bugs attacking the calf. So first of all is the cow, of course, parity, the quality of the colostrum, any health events or any problem during the calving of the cow. We also know that environment plays an important role when it comes to bugs attacking calves, so that does not depend on us. So season, weather, wet conditions, or any extreme climate event will also impact the health of the calf. The individual characteristic of the calf, so its genetic immunity, they will all be different. Maybe most importantly, things that we could manage. So the colostrum, the time when we give the calf the colostrum, how much we give it, if we're giving good quality. Biosecurity practices, so making sure bugs that are very bad don't get into the farm. Of course, the nutrition, the hygiene, making sure the bedding is clean and dry. Separation of the sick animals from the healthy ones. And then if we keep the calf strong, we might keep it safe from a lot of the bugs that we found. So about the study, the reason we did this study is because there might be unknown bugs that we're not yet aware of that are impacting the health of our animals. So just to give a background of why we're doing this research, we usually, when we have a sick animal, we take a sample, the vet will take a sample, and it will ask the vet lab to test for a bug, depending on the clinical presentation of the disease. So the vet might say test for Mannheimia, test for pestivirus, test for Moxella, depending on the clinical signs. And then the lab will just test for that. The traditional way in which we do it is we just test for what we think it might be there. And a lot of the time, we get an answer. So we get, it's Mannheimia, so there's things we can do about it and problem solved. But a lot of times, the results come negative, and it might be that we didn't get the correct sample, or it might be that there are bugs that we don't know of. And the newer research methods that we have, they look into all this bug that might be escaping our diagnostic test. They do look at all the genetics that are inside a sample, and with those genetics are the microbe ones. So we can detect everything that's in a sample without first saying test for this or for that. The sample will tell us what's there. So B2F project in 2023, we drove around New South Wales and we visited several of your farms. We did a lot of swabbing. We sampled 918 calves. We went to the far north, Hunter, far south, inner west, Riverina. So we try to do a representative samples of all the farms in New South Wales with this sampling strategy. We ended up doing 411 total profiling of these bugs of the nasal swabs and 595 of the rectal swab. We also sample cows, but that's a different presentation in a different study. So from those, we have animals that we sampled and we profiled all the ones that had respiratory disease, all the ones that had diarrhea, But we also sampled the healthy ones because we wanted to see what was going on the healthy animals, and we wanted to be able to compare what was going on in the sick and compare it to the healthy. So straight into the results. With respect to viruses, the most common that we found in those nasal swabs were this here. So the number that you see, there is the number of farms that were positive. And for a farm to be positive, is at least one animal positive to a virus. When you start looking at this list, you see many viruses that you might not have heard before. So Cobovirus, picornavirus. There's the bovine version of many viruses that you might have heard in humans. So bovine norovirus, adenovirus, rhinitis. People with allergies might have tested positive to human rhinitis, for example, virus. And then only in the sixth place, we have a bovine coronavirus, which we might be a little bit more familiar with. And at the very bottom, few farms with bovine respiratory syncytial virus and parainfluenza, which are usually in our first list of the ones that we might suspect that might be causing issues. Now, when we do the aggregated level and we compare the sick with the healthy in the 72 farms, we find that at the 2 week of age and 3 to 5, we find bovine rhinitis A very strongly associated with those animals that were sick. We also find coronavirus and adenovirus moderately associated with disease. That doesn't mean that, for example, bovine respiratory syncytial virus or parainfluenza could be a problem in a specific farm. But when we look at the industry level and the whole numbers that we have, we find that bovine rhinitis might be a good target to follow up as research. In terms of bacteria, it's different. So the ones that we find at the very top, most of the farms have this bacteria was Mycoplasma, Moraxella, and Mannheimia. So Moraxella causes, it's usually associated with pink eye. Mannheimia, we know that there's a vaccine, so there's things we can do against that. And when we look at the association with disease, we find Moraxella and Mannheimia mostly associated with that disease, and other bacteria that we need to look more into. The good thing is that against this, we do have some vaccine. And for Moraxella, in the next steps, we are looking into also understanding the vaccine efficacy. In terms of the rectal swabs, now the enteric viruses, a lot of the farms were positive to many of these viruses. So all of the farms had at least one animal with rotavirus A. They also have a lot of rotavirus and all other viruses that you might also not be very familiar with. When we look at the association now with disease, we find many of these viruses that we don't know really well how they are causing disease, but we found it very associated with those animals that are sick. Rotavirus in the second week and 3rd to 5 week of age. Rotavirus is one that we know. We're not really sure how the vaccine is really reacting to the viruses that are present in Australia because the vaccine needs to match the field virus. But we do find a lot of these viruses that we need to do a lot of research on to understand how they are causing disease. In terms of the bacteria in the gut, we didn't compare the sick and the healthy in terms of the type of bacteria because we know that there's a lot of healthy bacteria and there's a lot of disbalances. But when we look at that genetic information that we get in the sample, there are genes that make those bacteria attack the calf. And when we look at those genes that are expressing this bacteria, the bacteria that are expressing most of these bad genes are E. coli and Campylobacter. Now, E. coli is easy to grow and easy to detect in the lab, but Campylobacter is hard to grow in the lab. So it might be that that technical issue is making us underdiagnose these bacteria causing diarrhea in cows. In terms of parasite, crypto was the one that was most associated with sick calves. But we also found Giardia, Entamoeba, and Emeria. So Emeria is Coccidia. In terms of this association, first week of age, crypto was definitely the strongest associated with it. But in the second week, we found this in Tamoeba, which we really need to look more into because we weren't expecting that. We think that this is more not a very pathogenic bug, so that's something that we need to look more into. I didn't put the results of pestivirus, but as Ian mentioned and as Andrew mentioned, an outbreak that was causing issues in one of the farms. 17 farms were positive, and this is likely just the tip of the iceberg. We know that pestivirus is a major driver of disease in calf, so not only pneumonia, but diarrhea, infertility, and once it's there, it can cause a lot of trouble. It can be preventable through vaccines, and once it's important because if you know that your farm is negative, you have to be very careful not to get sick animals or positive animals into your farm. For all those viruses and all the bugs that you found in this study, we sent the results back that you don't know a lot about. We created this fact sheets to give you information about what's known, what's the current research about those bugs. So you can go into the Dairy App website. Also, you have some in the journal, the book, the Dairy App book, with information about these bugs. So the main achievements for this project is that we do have a clearer picture of calf disease. We are looking into that genetic mapping. So we're looking into the specific genotypes of pestivirus, the specific genotypes of rotaviruses, and that helps improve our diagnostics and vaccine development. And we know which bugs to look for in terms of the next stages. So the case is not closed. We can't say that this bug actually caused disease because a correlation scientifically is not causation. That means that we need to look more into this bug. To prove that a bug causes disease, we need to grow the bug, we need to put it in an animal, and we need to make that animal sick and replicate the cycle. That has not been done for all of the bugs that we are finding. So we are looking into trying to investigate that, to look into the organs of the animals that come into the lab and see where those bugs are located to see if they're really causing disease. So with that, I'd like to thank a lot of people, colleagues at EMAI, UTS. Zain has done a lot of the work extracting those samples and doing part of the data analysis. DRF, of course, Cybus, and all the team. LLS vets who helped a lot with the samplings. Jane also who helps us a lot visiting the farms, giving the results back. And most importantly, all the farmers who welcome us into their properties and supported this work. So thank you very much.
Barbara, thanks very much. It's pretty rare, folks, to see such a large epidemiological study in calves being presented anywhere in the world, to be frank. So, Barbara, congratulations. Questions? There's got to be some questions on this grand venture. Jenny, let's go with you first, please, at the back.
Thanks, Barbara. Really interesting presentation. I'm curious to know what you're doing in the vaccine space. So you've touched on that a little bit, and especially whether you're venturing into the RNA vaccine area.
Yes, that's a very good question. So in terms of rotavirus, there's contradicting evidence of how effective the rotavirus vaccine is. And that's because it's made with strains from overseas, or the strains might not match the field strain that we have here, the type. If you look at the rotavirus, it says it's against G6 and G10 genotypes. That's not the whole story. We do have mostly G6 and G10 genotypes. We do want to do a vaccine matching study to see in vitro and in silico if it matches, because we're not seeing the results of the vaccine that we would expect, and the literature has very contradicting evidence. So we really don't know if that's working. So that's one of the vaccine efforts that we're doing. We're also looking into a molecular epi study of Moraxella that will also help develop vaccines, but that's with the Sydney Uni group with John House.
Malcolm.
Actually, my question's around the last bit of what you talked about around, I guess, injecting some of the, or making some of these animals sick and the efficacy of that, and being involved with the project with Andrew. And what was it, Andrew? Who administered the meloxicam? How much did you administer it? Under whose supervision did you administer it? And yeah, we'll get into the very short texts as the people in Andrew's project was trying to drill down how come this person was administering meloxicam, and it really hit me as to how difficult doing something like this could be, but could actually be revolutionary in actually further healthcare or better healthcare for our animals into the future. So I guess, how do you get something like that up and running where it could have massive animal welfare benefits, but we do need to actually make some animals sick because, like I say, it was horrendous, wasn't it, Andrew? There was about 4 or 5 different text exchanges, and my blood pressure was going up every time some idiot in Sydney Uni wanted to know something. Hopefully they're not in the room.
Yeah, that's a very good question. So experimental work with animals is something that's very controversial. We haven't done anything. That's why I like epidemiological observational studies, because we just go and measure what's there and try to do the correlations. Now, just to try to understand, and in the sake of science, to prove that a pathogen can cause disease, few trials need to be made to understand that. But the more observational data we have, the better. I think that, that's the way. The more data we have, the less animals we have to use to understand that pathology. But in terms of that experimental work, it cannot be done with all of the bugs. So some bugs affect the animals very acutely, so it's really easy to put in there, the animal gets sick. Some bugs act in a very chronic way, so it's very hard to do that kind of experiment and understand how it works. Some bugs need other conditions to cause disease, so that's not just the bug put in the animal. So yeah, I think observational studies are a very good way around that experimental work with animals. Yeah, I agree. It doesn't feel good to experiment with animals.
Thanks. Well, I'm going to give the last question to this young Argentinian gentleman in the front. Oh, yeah, good joke.
Barbara, a really nice work and really cool data set as well. Just curious to know if with that data set you're able to look at the interrelationship between bugs, given that sometimes one bug is not pathogenic but opens the door for
Yes.
pathogen.
Actually, Zain is looking into that. Yeah, there are some interesting correlations. We're looking into, like, we're able to see some bugs and some specific of those bad genes. So with E. coli and Campylobacter, but that's something that's currently being researched, the interaction between bugs. Yeah, we have very good data to test for those correlations. So
Looking forward to those results.
Folks, can you please thank Barbara again? So I'm going to hand back to Paul. I'm just very thankful that the disco lights didn't go off during any of the presentations. I take that as a good sign. Paul, over to you.